This is the 11th video on the fundamentals of biology, and today we're talking about DNA and DNA replication. Here are some scientists that we need to know regarding DNA. So James Watson, Francis Crick, and Rosalind Franklin helped to discover the double helix structure of DNA, and Rosalind Franklin used something called X-ray crystallography, which is the first time we were able to get a picture of DNA. Frederick Griffith discovered that when pathogenic cells were heat killed, the remaining bacteria were able to assimilate into foreign DNA, and he called this process transformation. The importance of this is that DNA can be injected into other organisms. Alfred Hershey and Martha Chase used something called T2 bacteriophages in order to discover that DNA, not protein, was the genetic material. And they did this by first using radioactive sulfur to track protein, but they realized that it was not passed on through viruses. However, radioactive phosphorus traced DNA, and they saw that it did pass on to a bacteria. Finally, Erwin Chargaff discovered that DNA composition will vary within each organism, and he also discovered that within each organism, the adenine and thymine quantities or percentages will be equal, as well as the guanine and cytosine percentages. So this is the structure of DNA. First, you can see that it has a double helix structure, and the main lines here are sugar phosphate backbones, and they are anti-parallel. So this one on the left side will go from 3' to 5', whereas the one on the right side will go from 5' to 3'. Each nucleotide will have one of four nitrogenous bases, being G, C, A, or T. And a purine will always pair with their respective pyrimidine, and A and G will always be purines, and pyrimidines will be always T and C. And A's will always bind with the T's, and the G's will always bind with C's or hydrogen bonds. DNA replication is usually called semi-conservative. This is because when DNA replicates, each of the daughter DNA strands will have one old strand from the parent and one newly created strand. So you can see in this middle diagram showing semi-conservative, you can see that after the first replication, there will be one newly formed strand while there will be one old strand, and this will continue on to the second replication as well. This contrasts conservative, where two parent strands will rejoin, and this also contrasts dispersive, where each strand will have a mix of the old and new DNA on each strand individually. So conservative and dispersive are wrong, whereas semi-conservative is correct. So this is the beginning of DNA replication. A protein called helicase will unwind the DNA at the replication fork by breaking hydrogen bonds between nitrogenous bases. So this blue thing over here is the helicase, and it unwinds the DNA. Single-strand binding proteins will prevent the DNA from shutting back together, so these purple circles will keep the DNA from shutting. An enzyme called primase will create RNA primer, and primer is a short stretch of nucleic acids, and they will go on the 3' end. So this primase will create an RNA sequence that is short, and what this does is it allows DNA polymerase 3, also known as DNA polymerase, to add on to this 3' end using DNA nucleotides. Because DNA is double-stranded, there will be a leading and lagging strand. Because polymerase can only make DNA from 5' to 3', the strand that is being made 5' to 3', which is complementary to the 3' to 5' parent strand, is called the leading strand and is continuously synthesized, because after the primer is made, the DNA sequence is able to smoothly create more nucleotides. However, the strand that is 3' to 5' is called the lagging strand, and it will require many primers, and this will cause Okazaki fragments to be formed. Here are some other proteins involved in DNA replication. The sliding clamp will hold the DNA polymerase 3 in place. So this is the sliding clamp over here. Next, the topoisomerase will prevent the double helix ahead of the replication fork from becoming too tight by unwinding the DNA. So this is topoisomerase. Next, DNA polymerase 1 will replace the RNA primers after they are not in use anymore. DNA ligase will seal fragments together, such as Okazaki fragments in the lagging strand. Now let's talk about telomeres. In linear DNA, the replication of 5' ends cannot be completed, so every time DNA replication occurs, they will grow shorter. As a result, the cell will require telomeres, and these are special nucleotide sequences at the end of the eukaryotic DNA, and this will postpone their erosion at the end. Now let's talk about the coiling of DNA. Usually, DNA is unfolded and combines with proteins in order to become something called chromatin. On top of the chromatin, there will be beads called nucleosomes that will package together. Histones, which are found on the nucleosomes via histone tails, will help pack the chromatin together into chromosomes during interphase. Loosely packed DNA is called euchromatin, and this means that the cells will easily be expressed within the gene. However, tightly packed DNA is called heterochromatin, and this means that genes are difficult to be expressed. So thanks for watching. This lesson covered DNA discoveries and structure, DNA replication, and DNA packing.